When they enter a fish body, they bring alteration in internal structure of fish. So Clarius caripinus are taken as a model for ecotoxicological studies because of these reasons. They have voracious feeding habit, general alertness in water environment, and they are easily adjustable to laboratory conditions. So my general objective was to study the effect of cytometrin in air-breathing catfish, Clarius caripinus. The specific objectives were to determine the behavior of fish, to assess the effect of cytometrin in histology of ovary and testis of fish, and to calculate gonadosomatic index. Significance of the study: indiscriminate application of pesticide poses a noxious threat to non-target organisms and it disturbs the ecosystem. So this study was done to assess the limit of toxicity by using standardized protocols to protect the environment from harmful effect of hazardous chemicals and to protect the aquatic biodiversity from the effects of manufactured chemicals and helping to maintain the aquatic ecological niche and succession. So these are the materials which I have used during my study period. Cypermethrin the chemical cypermethrin was purchased from local agricultural shop. For animal maintenance, catfish fingerlings were procured from Kalimati Fish Market and they were brought to laboratory of Central Department of Zoology. Then they were treated with 0.2% of potassium permanganate for 1 minute to remove an external infection, and they were acclimatized for 25 days in a glass tank of given dimension, having capacity of 103.7 per liter. At the given temperature, where only 65 liter was used for acclimation. Fish in the given weight and length were used for the study. They were fed with chicken liver, and yes, water was changed daily, and cycling of uneaten feed and fecal matter was done. Yes, similarly, the physicochemical parameters of water was also assessed. For pesticide preparation and experimental design, 3 sublethal doses were taken from that is 0.008 ppm, 0.015 ppm and 0.02 ppm from the LC50 value, that is 0.21 ppm, that is parts per million of cypermethrin. Each concentration was given using micropipette, and fishes were starved for 24 hours before starting the experiment, and they were grouped. They were divided into 4 groups of 20 each, consisting E0 as a control group, and E1, E2 and E3 were pesticide treated groups. This was the experimental setup for different concentration of pesticide. During the experiment, fishes were fed 3% of body mass and mortality was recorded. To prevent oxygen depletion, experimental tanks were provided with air pump, and yes, dead fishes were removed immediately to avoid deterioration of water quality. Water was renewed daily, and concentration of cypermethrin was maintained for 96 hours. After 96 hours, the organs after 96 hours, fishes were sacrificed and the organ, ovary and testes were removed for further histological observation. Gonadosomatic index was calculated by the formula weight of gonad by weight of fish into 100. These are some of the pictures showing length, weight measurements and dissection of fishes. For histological observation, the tissues were fixed in Bowen solution, dehydrated with alcoholic series, cleared with xylene, blocked with paraffin wax, sectioned with microtome, fixed in microslide, processed for dewaxing using xylene, hydrated with alcoholic series, stained with hematoxylin and counter-stained with eosin, dehydrated with alcoholic series and then mounted with DPX. They were observed in a microscope, photographs were taken, and the comparison between control and treated was done. Treated tissues were done. These are some of the pictures showing the procedures which I have followed during slide preparation. I was helped by Anita Jonche ma'am, who is the lab technician at Animal Health Research Division for microtomy and staining procedure. Statistical analysis were performed in Microsoft Excel 2016. So moving to results, morphological and behavioral changes. So the fishes which were in control medium, they showed normal swimming patterns. Their color was not changed, it was dark pigmentation and fin movement during the exposure period. After 15 minutes of pesticide introduction, treated fishes exhibited vertical and horizontal swimming patterns, erratic swimming, hyperactivity, tail chasing behavior, nipping each other's fin, leading to wounds mainly on dorsal parts. Later they showed characteristics like dullness, low food capturing, increase in gill mucus secretion, bluish coloration in ventral part of some fishes, and stiffening of the body was also observed. Similarly, the color of fish skin faded from dark pigmentation in the dorsal and lateral parts to very light. Sorry. Where are my pictures? Huh? I'm sorry for that. There was the picture showing bluish coloration on ventral part of fish, and due to the pesticide exposure, the fishes started to like nip on each other's body parts, and different bruises were also observed in their body. I'm sorry for that. Okay, next, moving to fish mortality. Highest mortality was observed in E3, that is 0.02 ppm. That is the highest concentration, and no mortality was observed in E0, that is the control medium, and E1 24 hours. Similarly, figure 1, it shows mortality percent of treated and controlled fish at different pesticide exposure with the change in duration. So from this graph, it shows us that as the pesticide concentration increase, the mortality in fishes also increase. So what we can know that the mortality of fish is concentration and pesticide exposure dependent. So table 2 shows gonadosomatic index GSI values of both male and female at different concentration of pesticide. Figure 2 shows effect of cypermethrin on testicular and ovarian GSI of Clarius caripinus exposed for 96 hours. So from the graph, we can see that this E0 is the control medium, which shows the highest GSI value in comparison to this treated GSI value. So this shows, and what we can know is from this graph, female GSI, these all are female GSI value, which is higher than male GSI value. So we can know that this pesticide cypermethrin has more effect on female ovary. Okay, next, histological observation. So in control ovary, it showed normal ovarian development with oocyte wall enclosing chromatonuclear stage oocytes. Small size oocytes are seen because it is a fingerling. So immature stage oocytes were observed, and yes, yolk was evenly distributed. When ovary was treated at 0.018 ppm, it showed cell adhesions, cytoplasmic clumping. Likewise, when the ovary was treated at 0.015 ppm, it showed cell adhesions, degeneration of follicles, vacuoles, degeneration of oocyte wall, necrosis, and atretic oocytes. At the highest concentration, treated ovary showed vacuoles, adhesions of cell, atretic oocytes. This one is a continuation of that same treated with 0.02 ppm. It shows vacuolations, and yes, large number of immature oocytes can be seen. And in this picture E, we can see complete degeneration of oocyte wall. Moving to testis, control testis showed normal testicular arrangement having germinal epithelium, spermatids, primary spermatogonia. And when the testis was treated at 0.008 ppm, it showed disintegration of spermatogonia, ruptured wall. Likewise, when it was treated at 0.015 ppm, it showed dilation of seminiferous tubules and condensation of spermatogenic cells. At highest concentration, that is 0.02 ppm, it showed degeneration of seminiferous tubules, vacuoles, fibrosis. Other changes were also observed, so which were more highlighted, I have mentioned over here. So the conclusion and recommendation which I have obtained from my current study: the fish exposed to the toxic in cypermethrin showed abnormal behavior like loss of equilibrium, hyperactivity, jerky movement, which is an indication of degenerative changes in tissue level. As the pesticide exposure increased, deformities in ovary and testis were highly observed. The treated fish ovary showed cytoplasmic vacuolation, clumping of oocytes, atretic vitellogenic oocytes, and yolk globules which became more lipidous, that means they became more fatty. And the treated testis showed disintegration of spermatozoa, rupture of testis wall, dilation of seminiferous tubules. Though cypermethrin is established neurotoxic, it shows its adverse effect on gonads. Maybe it affects the secretion of FSH, that is follicle stimulating hormone. It regulates the function of ovaries and testes. Moreover, GSI is very useful matrix for assessing fish health status, which provides knowledge on determining the deformities in environment. This work can be used as a baseline information for further studies and suggests that Cladis caripinus is an important bioindicator animal for evaluating and monitoring the effects of toxicity in water bodies. Hence, additional studies on biochemical analysis, because this histological observation is not enough. Biochemical analysis on effect of cypermetrin are still needed to fill the research gap and greater perception into the mechanism of cypermetrin. Finally, I would like to acknowledge my respected supervisor, Associate Professor Dr. Archana Prasad, our Head of Department, Professor Dr. Tej Bahadur Thapa, Professor Dr. Kumar Sapura for helping me in title selection, Professor Dr. Suryaratna Gubaju for fish identification, Lecturer Santoshi Shrestha for her support during the entire study, Mr. Swayam Prakash Shrestha, Chief and Animal Health Research Division, for providing me the lab to perform my work, and Mrs. Anita Jonche, who is the lab technician at the same place, for assisting me in that whole microtomy and slide preparation process. Professor Dr. Upendra Mansingh, he is the head of department at HICAST, for his valuable suggestion regarding histological observation. Thank you everyone for your time and presence.